What is plant tissue culture? Plant tissue culture is a technique used to propagate plants by growing them in sterile laboratory conditions. The process involves the growth and multiplication of plant cells, tissues, and organs in vitro, or outside the plant's natural environment, under controlled environmental and nutrient conditions. It is used in various fields, such as agriculture, horticulture, forestry, and research. The concept of plant tissue culture was first developed in the early 20th century when it was discovered that plant cells could be grown in vitro. Since then, the technique has evolved and become an essential tool for plant biotechnology. It is used to propagate plants from a single cell, create disease-free plants, and produce large numbers of genetically identical plants, known as clones. In addition, plant tissue culture can be used for genetic transformation, regeneration of plants from single cells, and the production of secondary metabolites. The process of plant tissue culture begins by obtaining explants, which are small pieces of plant tissue, from the desired plant. The explants are then sterilized and placed onto a sterile nutrient medium containing growth regulators and other nutrients. The nutrients are tailored to the specific needs of the plant tissue being cultured. The tissue is then incubated under specific temperature, light, and humidity conditions to encourage growth and development. The type of growth regulator and nutrient combination used depends on the desired outcome. For example, the hormone auxin is used to induce root formation, while the hormone cytokinin is used to promote shoot development. The optimal balance between these hormones is critical for the success of plant tissue culture. The culture vessel used for plant tissue culture is also important. The most commonly used vessels are test tubes or PETRI dishes with a nutrient medium. These vessels are sealed with a sterile lid to prevent contamination from airborne microbes. The culture vessels are then placed in a growth chamber with controlled environmental conditions such as temperature, humidity, and light. Once the tissue begins to grow and develop, it can be subcultured to produce multiple clones. The process of subculturing involves transferring a portion of the growing tissue onto a fresh nutrient medium to continue growth and development. By repeating this process, large numbers of genetically identical plants can be produced. Plant tissue culture is used in various applications such as micropropagation, which is the production of large numbers of identical plants, as well as the production of secondary metabolites such as alkaloids, essential oils, and pigments. It is also used to develop disease-resistant plants, to study plant genetics and physiology, and to genetically modify plants for improved traits such as yield and quality. In summary, plant tissue culture is a powerful tool for the propagation of plants in vitro. It allows for the production of large numbers of genetically identical plants, disease-free plants, and the development of plants with improved traits. It is used in various fields such as agriculture, horticulture, forestry, and research. The process involves the growth and multiplication of plant cells, tissues, and organs in a sterile laboratory environment, under controlled environmental and nutrient conditions. The technique has revolutionized the way plants are propagated and is expected to play an even more significant role in plant biotechnology in the future.